Welcome back, everyone, or welcome if this is your first session at the 2020 Layman Conference. We are excited to have you with us today. Those of you who have been noodling through the website, I'm happy that you're back and hope you've had a chance to look at our sponsor links and gone through those. There's a lot of good information in there. A little harder to find is the donation to the the Second Harvest Heartland food donation spot. If you click on the More button, we're up to over 17,000 meals that have been donated so far. So please take advantage of that if you have a chance. And I also hope you've taken advantage of the flu vaccine pledge. That's something that we have a strong history of here at the Layman Conference, and obviously it's a very important part of how we take care of ourselves, our staffs, and our pigs. So very good. So welcome to our coronavirus session. Obviously it's a big issue for us this year, and so we've added this in as a special session. Pig veterinarians are coronavirus experts by nature because we all have quite a bit of experience with 5 of the 6 coronaviruses that affect pigs. And so we thought we would build on our long suit and include some speakers that can provide some other perspectives as well. So we have 3 presentations today. The first presentation is by Dr. Linda Saif from The Ohio State University. She is a preeminent expert globally on coronaviruses in particular. I first met Dr. Saif a while back. I don't know, Linda, if you remember, we invited you to come and give a talk when there was a new virus introduced into North America, not PED, not SARS-CoV-2, but porcine respiratory coronavirus. And that was 25 years ago, and you were already 15 years into being an expert on coronavirus at that time. She was invited into the U.S. National Academy of Sciences in 2003. And from my perspective, that pretty well says it all about her tremendously distinguished career as a researcher and helper for our industry and the SARS episode a few years back, and now SARS-CoV-2. So without taking more of your time in your presentation, Dr. Saif, we are thrilled that you're willing to be one of our keynote speakers today, and welcome to the Layman 22 conference, or 2020 conference. We look forward to your comments, and we'll take questions at the end of her presentation. So please keep the chat room busy with the participants. Thank you.
Okay. Well, thanks, Jerry, for the nice introduction. I'll try to bring my slides up right away to stay on time. And I've been to several of the Layman conferences, as Jerry said, some time ago for the one of them. And today I'm going to talk about emerging coronavirus of both humans and animals, and several aspects, origins, interspecies transmission, and vaccines, with a focus on COVID-19 and swine coronas. So for those of you who aren't familiar, the Coronaviridae family has 4 genera, the alpha, beta, gamma, delta. They're positive sense envelope single-strand RNA virus, the largest RNA genome virus, and they have these spikes on the surface, shown schematically here in green. And this spike gene encodes the virulence determinants and the receptor binding domain to attach to the host cell receptor. And it's also then the target for neutralizing antibodies. They infect mammals and birds, and they cause mainly respiratory and enteric disease. So the COVID-19 that we're all very familiar with now emerged first in China 7 years after the Middle East respiratory syndrome in Saudi Arabia. It was first detected or reported in December or November 2019, a large outbreak in Wuhan, China. It was rapidly sequenced. It was named as SARS coronavirus 2 as a new beta coronavirus B, but it was most closely related to the SARS 80% nucleotide identity, and also closely related to one strain of bat coronavirus, 96% overall identity. At the end of January, WHO announced the virus was a public health emergency of international concern. And we're all aware it's now a pandemic with over 31 million cases worldwide, almost a million deaths in 216 countries. COVID-19 spread to the US in January, February of this year. And as of today, we have almost 7 million cases, almost 200,000 deaths in 50 states. So this slide depicts the coronavirus ecology and the diverse hosts. We have the alpha coronavirus here that consists of viruses that infect wildlife, domesticated animals, companion animals, and these are thought to have a bat ancestral origin. And we know that within the TG group, there are host range mutants, the canine coronavirus, the TG and PRCV and FIPV, and related viruses in cats. And these still interchange and can cause interspecies infection among these species. And this gives rise to new strains of viruses. We also have in this group the 229E, the common cold coronavirus that was thought to be transmitted by bats to camelids to humans. And then in the next clade, also bat-derived coronavirus, or the beta coronavirus, the clade A. Again, many species, including domestic animals and rodents, as well as wildlife. And it's thought that the common cold OC43 was transmitted from cattle to humans historically. And then we have clade B, the original SARS, from bats to civet cats to humans. And then the recent SARS coronavirus 2, presumably from bats, either directly or indirectly to humans. We don't know if there's an intermediate host. And clade C, the MERS coronavirus, from bats to camels to humans. Now, the gamma coronavirus are mostly avian derived, and so are the delta coronavirus, but there's been some spillover of these delta coronavirus from avian species to mammals, including swine, with an outbreak in the US in 2014. So we now know there are 7 human coronavirus, mainly associated with respiratory infections. The endemic ones cause the common cold. The population has variable degrees of immunity, but this immunity is projected to last only about a year from experimental human volunteer studies. So the alpha coronavirus are 229E, NL63. The betas are OC43, HKU1. And then we have these emergence of these epidemic, pandemic strains in a naive population with no immunity. And these are the beta coronavirus, SARS, MERS, SARS coronavirus 2. So for the swine strains, we have the endemic gastroenteritis strains, the alpha coronavirus PDV and TGE, endemic respiratory strain, the derivative of TG, the respiratory coronavirus, PRCV, and then another endemic one, encephalomyelitis, hemagglutinating encephalomyelitis, which is the only beta coronavirus. And then epidemic diarrhea strains in a naive population with no immunity. Again, I'll talk more about this, the emergence of the alpha coronavirus severe acute diarrhea syndrome in China, and then more recently also the porcine delta coronavirus. So most human coronavirus belong to beta coronavirus genus, and most of the swine coronavirus belong to the alpha coronavirus genus. So what I'd like to talk to you about today are a number of questions. Why are the coronavirus so diverse, and how do they emerge and evolve to infect new hosts? Which coronavirus cross the species barrier? What are the animal reservoirs for SARS and COVID-19? How are they transmitted from animals to humans? How does SARS coronavirus 2 cause disease compared with swine respiratory coronavirus? And then the last part, the unknowns or gaps for SARS coronavirus 2 in vaccines, correlates of protection and lessons for immunity from some of our swine coronavirus. So the first question, why are the coronavirus so diverse, and how do they emerge and evolve to infect new hosts? The coronavirus have a very large RNA genome, as I mentioned, and even though the viruses have evolved a proofreading mechanism, the polymerase still has a relatively high error rate, so mutations are common. Coronavirus occur as a quasi-species or a swarm of viruses in a population. So when you're infected with a coronavirus, it's not a single coronavirus, it's actually a quasi-species of coronavirus particles. And this may explain the origin of interspecies variants with altered tissue tropism. And the one I'm going to talk about is the swine coronavirus, the enteric strain TG, and then the respiratory variant PRCV. But we also have examples with feline coronavirus, an enteric strain, feline enteric coronavirus that mutates within the cat to cause a fatal disease, FIPV, feline infectious peritonitis virus. So coronavirus also have high recombination frequency, and this can lead to the emergence of new strains or new zoonotic strains. So early on, we wanted to examine the question whether there were coronavirus in wildlife, and also if the coronavirus then could cross the species barrier. So this study was done by a postdoc in my lab, Dr. Tsunemitsu, in 1995, and another postdoc, Dr. Han, who worked with the strain from a child in 2016. And we isolated these viruses from captive wild ruminants, sambar deer, white-tailed deer, waterbuck, and we also got from Dr. Zhang the strain isolated from a child with diarrhea, and this strain had a very similar sequence homology to the bovine coronavirus. So we used germ-free calves because they don't have any other coronavirus or existing microflora. And we inoculated all the calves with each of these different isolates. The viruses caused diarrhea in all the calves. All the viruses were shed by the calves fecally, and all of the calves were converted with neutralizing antibodies to bovine coronavirus. So this told us early on that indeed coronavirus are not necessarily host specific, but the coronavirus from wild ruminants or humans can experimentally infect young calves. So there's a ruminant coronavirus reservoir community for these clade A beta coronaviruses. So this slide summarizes something about the spillover and emergence of new coronavirus that has occurred repeatedly, both in humans and livestock. So we had the original MERS coronavirus from bats to civet cats in 2003 in China. And then about a decade later, we had the emergence of MERS coronavirus from bats to camels in Saudi Arabia. This is still ongoing. And then the most recent human pandemic strain, the SARS coronavirus 2, either directly from bats or through an intermediate host. SARS coronavirus, however, was not the first coronavirus transmitted from animals to human. And in fact, these coronaviruses associated with the common cold are endemic in humans. And what's not well appreciated, these are also zoonoses. So several centuries ago, we had the spillover from bats of the NL63 to humans, associated them with the common cold. In the 17 to 1800s, the spillover from bats through camelids of the 229E. And then in around 1898, and this is all projected by sequence analysis and molecular clock analysis, the spillover of the OC43 common cold from cattle to humans. So an interesting feature is that several of these viruses that have spilled over into humans, the intermediate host has been a ruminant. So we now know that coronavirus can have broad host ranges. Certainly the beta coronavirus, the SARS coronavirus 2, the SARS clade B, the MERS clade C, and bovine coronavirus clade A are promiscuous. We know now SARS coronavirus 2 can infect humans, related viruses in horseshoe bats and pangolins. And we know from that several cases now that the virus can spill over in infected households to cats and dogs, and some spillovers in the zoo and tigers and lions. And then I'm going to talk more about this, the spillover into mink farms initiated through humans. Experimentally, the virus can infect hamsters, ferrets, non-human primates, mice, rabbits, and tree shrews. We see a wide range of hosts for MERS coronavirus, including the camel intermediate host, and then same way for the SARS, the original SARS coronavirus, including swine can be infected with the SARS coronavirus 2 and also with MERS. And then a wide range of hosts for bovine coronavirus, including spillover into dogs and some avian species. So then we can ask what factors influence the interspecies transmission of these enteric and respiratory coronavirus. Well, one is the exposure when we have close contact with infected animals, infectious secretions or fomites, and this can occur at the wildlife, domestic animal human interface, especially where there are bat reservoirs or bats sold in marketplaces, wet markets, live animal markets like here in Asia, where we have multiple species unsanitary conditions. With PDV, it's thought the virus might have come from China to the US through fecally contaminated feed sources or some kind of environmental contamination. Another factor is the stability and dose of the agent in the infectious material and the transmission route. It's possible there's a greater efficiency of combined aerosol and fecal oral transmission for these pneumoenteric viruses, for bovine coronavirus, SARS, and MERS, and potentially SARS coronavirus 2, because about 20 to 30% of the cases experience gastroenteritis. Another factor is the viral attachment and entry with the role of receptors and co-receptors. It's interesting that some clade A coronavirus, including bovine coronavirus, has an influenza C-like hemagglutinin acquired in a recombination event, and that's shown as these short particles on the bovine coronavirus virus surface. And then, like SARS coronavirus 2 also binds to the same receptor ACE2 but by a novel different mechanism. It shows higher binding affinity for the SARS than for SARS coronavirus 2. So maybe this might be one aspect of why it's so transmissible, as well as it has a new furin cleavage site in the S1 and S2 that may influence the transmissibility. So we know that SARS coronavirus was a zoonosis. The observations were that the workers in the live animal markets in China had antibodies to the SARS coronavirus, but no disease. So then researchers looked in the markets in China to see what animals might be carrying the viral RNA, and they found it in these mass palm civets shown here that had very high nucleotide identity to SARS, but not in the wild and farm civets. And then after the SARS outbreak, sequence data confirmed that certain bats harbor SARS-related coronavirus, and these are the rhinolopus bats or horseshoe bats. And indeed, a more disturbing finding was that some bat strains can actually use the human SARS coronavirus ACE2 receptor. So this suggests that bats could actually play a role in direct transmission. And this prompted a lot of coronavirologists to worry about future spillovers of SARS-related coronavirus from bats. And in one report, the scientists featured these SARS-like coronavirus in bats as a time bomb for the next pandemic, which we now know has materialized. So is 19 also a zoonosis? Again, the observation, some of the early cases, about 2/3 had contact with live animal markets in Wuhan. And so what is the host animal reservoir for SARS? Again, it's likely bat host. And an interesting observation was in Chinese researchers in 2018 surveyed for bats, SARS-like coronavirus in humans in southern China in the vicinity of these bat caves, and they found about 3% had antibodies. So that's evidence for prior spillovers. And then two bat coronavirus were shown to be closely related to the human SARS-2. The one strain, as I mentioned, shares over 96% identity, and a second strain shares high identity in the open reading frame 1A-B, the polymerase. But interesting, although the original SARS strain does not have this cleavage site, it also has an S1, S2 furin cleavage site. So is there an intermediate host that transmitted the virus to humans beyond bats? So some suspect the pangolin, shown here, because it has a high nucleotide identity in the receptor binding domain to the SARS coronavirus 2, but then more recent studies have suggested that the affinity for the human ACE2 receptor may not be so high. So potentially other infected animals in the wildlife markets. And one suspect I'm going to talk about more are mink, because actually China is the leading fur producer in the world. or a possible related mustelid species. So the next question is, could pets and mink serve as incidental or secondary hosts for COVID-19? Because we know there were cases where cats and dogs have been infected in household of COVID-19 positive cases. So then people did experimental studies of cats and dogs, and cats could be experimentally infected in 3 different studies. The cats shed virus, they showed variable respiratory pathology, they seroconverted and transmitted the virus to contact cats. However, the dogs were only marginally susceptible, but they did seroconvert. And then in a study in COVID-19 affected regions of Italy, they surveyed household cats and dogs, and they found almost 4% of cats and 3.4% of dogs seroconverted. But then if they looked in the households of the positive cases, this percentages were about 2 to 3 times higher. And another interesting aspect in terms of cats, um, in the studies of these infected mink in the Netherlands, when they surveyed stray cats, they found about 30% of the stray cats were seropositive for SARS-CoV-2 that were found in the vicinity of these infected mink farms. Now, to date, there is no evidence for the transmission, the reverse transmission from pets to humans. So the second concern here is for the mink, because there have been major outbreaks now in farm mink in the Netherlands, Denmark, Spain, and then recently in Utah in the USA. And it was shown there's rapid indirect transmission among these caged minks, so almost 1 million mink were culled. The mink showed similar symptoms to what we see with COVID-19 in humans. Some had mild to severe respiratory signs, they shed virus, some developed interstitial pneumonia, and some died, and most of the mink seroconverted. Now, the more worrisome situation with the mink, that in more detailed follow-up studies done in the Netherlands, although the original source of infection was thought to have come from humans to spill over to mink, there now are at least two suspect cases of transmission back from mink to humans. So this raises a concerning question of whether pets or wildlife, including wild muscelids, felids, or even non-human primates in the wild, become infected with these viruses and become a new reservoir host for COVID-19. Now, this is a topic I'm sure everyone will be interested in, and unfortunately, there are not a lot of studies in this area, especially for livestock other than swine. So investigators looked to see if swine could be experimentally infected with these COVID-19 because their ACE2 receptor shares a high similarity with the human ACE2 receptor, similar to the other species that are susceptible. So in 3 separate studies, study in the US and Germany and China, they took 5 to 8-week-old pigs, and in none of these studies could the pigs be infected, shed virus, or seroconvert, or transmit the virus to other pigs. However, a recent preprint study has just appeared in the literature from Canada, and they reported that pigs could show marginal experimental infections. They detected low-level viral RNA in one group of oral fluids and nasal wash of 2 pigs. 2 pigs were converted with very low or borderline antibody titers. And in 1 pig, the submandibular lymph node, they detected viral RNA and they were able to isolate virus at this one time point. However, the consensus of all 4 studies is that there was no viral RNA shedding in feces or nasal secretions. There was no pathology upon postmortem, and there's no evidence of any contact transmission. Now, some discrepancies in these 4 different studies are the SARS, they all use different SARS coronavirus 2 strains, different doses, different swine ages, different breeds. Some used SPF herds, some had high health herd pigs. So at this point, it may be important to conduct some additional pig studies and also For monitoring, it may be important to start conducting some serologic gold standard virus neutralization serologic field studies for COVID-19 infections of swine to be sure there's not a spillover. So in summary, 3 of these 4 studies showed no evidence for experimental infection of pigs. Now I'm going to talk next about some of the spillover of recent swine coronavirus, again from either probably bat or avian reservoirs. So we're familiar with the timeline for most of these historic coronavirus strains, including the emergence of the respiratory variant in the mid-1980s. But I'm going to show some evidence for recombination between PEDV that the virulent strains appeared in China in 2010 and then spilled over in the US in 2013. So recombinant between PED and TG, swine enteric coronavirus, it was called, was detected in Europe, and then spillover presumably from avian. The porcine delta coronavirus identified in Asia in 2012, spillover in the US 2014, and then the occurrence of the severe acute diarrhea syndrome coronavirus, presumably directly from bats into swine, and that was in 2016 or 2017, shown up here. So now we have these emerging endemic alpha coronavirus recombinants in swine. So historically, we had PED first circulating in Europe, and then highly virulent strains in China, and then spill over into the US of these Chinese strains with fatal diarrhea disease in baby pigs in the US. Again, very similar to SARS coronavirus 2, where we have a completely naive population. All the swine were naive, had no antibodies to PEDV in the US. So it was a fatal disease in baby pigs, and it's estimated that in a naive population, as many as 8 million pigs in the US died, and now the virus appears to be endemic. So there were sporadic outbreaks of PED subsequent to this first discovery in Europe. And in 2009, it was independently reported in Italy, also Germany, and Eastern Europe, was the appearance of a recombination between the TGE up here in the yellow and PED here in the red, with virus recombining in terms of having a PED spike, but the rest of the backbone was from TGE. So there were some reports of diarrheal disease in the field associated with these recombinant viruses. And then we have the emerging epidemic coronavirus in swine with new species of coronavirus. We have the porcine delta coronavirus, I mentioned, identified in Hong Kong, but not associated with disease in 2014, spillover presumably from birds to pigs in the US, because it previously had occurred mainly in avian species. We discovered with Dr. Bosch in the Netherlands, it uses the APN's receptor like TG to infect pig, chicken, and human cells. And then studies in our lab showed it can also marginally infect calves with, they seroconvert with, but with almost an abortive type infection, but also can infect, go back and infect avian species, turkeys, shown by Dr. Kinney. So this is worrisome that the virus can now affect different avian and mammalian hosts, and this makes us concerned whether Delta coronavirus could be the next emerging coronavirus even for humans. And then again, we had the sudden appearance of the swine acute diarrhea syndrome in China. It killed an estimated 24,693 baby pigs in southern China. And when the sequences were looked at with the sequences of bats in the area, this was an alpha coronavirus that had very similar sequence between the bat strains that were detected in the area and the strains that were detected in the swine. So this looked like it could have been a direct spillover from bats into swine in China. So to summarize this part of my talk in terms of coronavirus interspecies transmission, we have bird reservoirs for Delta Gamma coronavirus. There's a large feed bunk in the Midwest where you see all these Canadian geese congregating and potentially then spill over through, again, contaminated feed fed to livestock into possibly pigs. Bat reservoirs for the alpha and beta B and C coronavirus, and then ruminants or rodents for the beta A coronavirus. So the reason these bat and bird reservoirs are so significant, there are multiple species that congregate, they have a very high density, and they're migratory, so they can spread these viruses further distances. And a concern then is once these coronavirus adapt to a new host species, they may expand within the new host with interspecies transmission, like I showed you for I mentioned for porcine respiratory coronavirus and TG, and establish a new reservoir community. So to summarize this part of the talk, the coronavirus continue to evolve and emerge globally as a cause of fatal respiratory and enteric infections of young animals and humans. They're genetically diverse with frequent mutation recombination, and some of the beta coronavirus are zoonotic. The epidemiology and zoonotic transmission of SARS coronavirus 2 infections are unclear. And unfortunately, no vaccines have yet been developed for respiratory coronavirus in mammals, although there is a infectious bronchitis vaccine for poultry. So WHO has identified the potential pandemic with what they call disease X. And unfortunately, it appears to be not if, but when, because of these bat reservoirs for many coronavirus, there may be future coronavirus transmission to humans. So the last part of my talk, I want to address two additional topics. Um, how does SARS coronavirus cause disease compared with the swine respiratory coronavirus? What are the unknowns or gaps for SARS coronavirus 2 in vaccines in terms of correlates of protection? And are there lessons for immunity from some of the similar platform vaccines that are being developed for use in humans with some swine experimental coronavirus vaccines? So initially it's thought that COVID-19 was predominantly a pneumonia respiratory disorder um, with acute respiratory distress syndrome in severe cases, a cytokine storm. But now we know it's also associated with some nervous disorders, loss of smell and taste, and some systemic disorders, acute cardiac injury, diarrhea, coagulation dysfunction. Um, so the big question is which disorders are actually virus-induced versus which are immunopathology or the host response. And this is very important in terms of vaccines and treatments, understanding the pathogenesis. We also don't know in terms of SARS coronavirus 2 why there's enhanced disease in the elderly versus the young. We know that the elderly respond with a more pro-inflammatory response to infections, and they also experience immunosenescence or a less robust immune response. Why is there enhanced disease in these patients with these comorbidities, especially diabetes, heart disease, obesity, COPD? And why is there such prolonged recovery period for some patients? So these unknowns can influence the design of COVID-19 vaccines because it's important to understand the pathogenesis of SARS coronavirus 2. Is it strictly a respiratory infection or does it also infect the gut or these other organs? Is it strictly through upper respiratory tract infection with viral dissemination to the lung, the intestine, or other organs? So for this, there are a couple models for vaccines. It's known that the attenuated intranasal influenza vaccine induces local IG antibodies, but lower levels of serum antibodies, but this can prevent the upper respiratory tract infection. On the other hand, it's also known that use of inactivated intramuscular influenza vaccine that induces high levels of serum IG antibodies, these serum antibodies can transudate to the lung and prevent lung infection. As I mentioned, the question is, does disease result from the magnitude of infection or immunopathology or both? We don't know the correlates of protective immunity in terms of innate humoral or cellular immunity. And does immunity to reinfection occur after recovery? And if so, might it be a short term, similar to the common cold? And this was reviewed in a recent review I wrote about COVID-19 vaccines. So I want to go back to now discuss some similarities in terms of the porcine respiratory coronavirus infection, which is a mutant of TGE. So we know TGE causes fatal diarrhea in baby pigs, but in the 1980s, this spike gene deletion mutant of TGE with a large deletion in the N-terminal region of the spike here appeared independently in Europe and the US. Now, TG and PRCV both share the same receptor binding domain, the APN receptor, but the tissue tropisms are thought to differ because of the loss of this region that binds to sialic acid or gut mucins. So this virus cannot stay stable and infect the gut compared to TG. So there's a loss of enteric tropism and virulence. We also have noted a lot of similarities to SARS-CoV-2 respiratory infections. PRCV infects the epithelial cells of the upper and lower respiratory tract and type 1 and 2 pneumocytes. Most infections are mild or subclinical, so the pigs can have something like a walking pneumonia. And interesting, this has been reported when they did CAT scans in about as high as 50% of the asymptomatic COVID-19 cases. And this atypical pneumonia that we see in most of the PRCV infected pigs resembles the SARS-CoV-2 lesions. So we and others have used PRCV as a BSL-2 respiratory coronavirus model for COVID-19. Now, in terms of the lessons from swine coronavirus veterinary vaccines, we have the scenario where we know that if we give TG orally, especially the virulent virus, but multiple doses of attenuated TG or even PED vaccine, this will induce the gut mammary axis gut milk Ig antibodies, and these are a correlate in the milk of immune protection. So our initial studies identified this gastrointestinal associated gut mammary SGI SIGA axis. So vaccinating the mother to get gut protection, and then getting milk Ig antibodies to protect the suckling piglets, and the same oral vaccine in weaned pigs to induce gut IgA antibodies. Now, the interesting aspect of that is after PRCV has been circulating in the swine herd, we also showed that if you have repeated PRCV infections in different pregnancies in pregnant animals, pregnant sows, that eventually you can also induce a bronchus-associated lymphoid tissue mammary gland axis and get IgA antibodies in the milk. So PRCV has acted as a naturally occurring TG vaccine. So I spent a lot of my career working on TG vaccines, and then PRCV came along and pretty much solved a lot of the TG problems for us. Now, for COVID-19, there's an array of vaccines, and these are just diagrammed here. So some are inactivated or weakened with a traditional approach. They're viral vectors, replicating, non-replicating with 2 in phase 3. These are mostly adenovirus-based human adnos. There's one approved for the military in China and one approved in Russia. Then we have nucleic acid, DNA, and RNA with 2 RNA in phase 3. And then we have protein subunit and virus-like particles. So I'm going to mention some of the data from the swine experimental vaccines that have attempted non-replicating adenovirus RNA vaccines and protein subunit. So that there are not too many experimental vaccine studies with PRCV because it's not thought to be a major severe pathogen in swine, but a study by Calabat with in a lab from Spain with Dr. Inmanis showed if they took a human adeno5, which is being used for some of the COVID-19 vaccines, and they cloned the spike of PRCV into that backbone, and this was actually a replicating virus, but most are non-replicating. They gave a single dose oral nasally, and then they challenged with PRCV. They got very low titers of serum antibodies after one dose. They couldn't really do morbidity because there was no morbidity even in the controls, but they did get partial protection in terms of shorter shedding, but not complete protection against infection. Recent study with PED enteric using a human adeno5 vector with S1 PEDV. Again, there were no antibodies elicited pre-challenge, but there was a big booster effect post-challenge. They reported partial protection against morbidity, but little or no protection against shedding, maybe shorter duration compared to the controls. So in swine, these human adeno S vaccine given in one dose elicited only partial respiratory immunity, and for PEDV, this was marginal. So I think at least this shows multiple doses are going to be necessary, and whether it's going to be efficacious or not, we won't know till the larger phase 3 trials. So additional lessons from molecular vaccine for swine coronaviruses. The advantage of these and the reason they're being tried for the human vaccines, even though they haven't previously been licensed for in humans, is to provide a platform for rapid production of vaccines for new emerging diseases. So what they can do is produce RNA, DNA, or vector backbone constructs, and then just insert key antigens for the new viruses, and then apply their established manufacturing procedures. But as I mentioned, the question is, will these unproven vaccines be effective to prevent both disease and shedding or transmission for COVID-19? So we worked for a long time on a spike recombinant baculovirus express vaccine for TG, and we found it was ineffective giving it parental IP or sub-Q to naive pigs. But if the pigs were given one dose of oral attenuated vaccine first, it was effective in protecting the pigs as a booster vaccine. So in terms of PEDV and RNA vaccine, there's a viral replicon particle based on Venezuela equine encephalitis virus. This is the construct that carries the PED spike replicon RNA. So this will be taken up in cells, and it's non-replicating single cycle of RNA expression. So in the manufactured study, 1 to 3 intramuscular doses in pregnant sows generally showed low efficacy in terms of compared to the controls, only 14 to 22% less mortality than the controls. And in an independent study, the protection was even less compared to the control, only 3% above what the control was. So another study showed that this type of vaccine induced low milk virus neutralizing antibody titers on only 50% of the sows, and with the VN titer less than 80, versus 100% of the wild type PD oral infected sows had neutralizing antibodies in milk and higher titers. So the next thing I want to discuss is strategies used to generate safe attenuated coronavirus vaccines using reverse genetics with infectious clones. So Dr. Wong has been pioneering studies with several graduate students and in collaboration with my lab, with a graduate student first with Jacob Hu, who did the study I'm going to mention here. So this involves targeting genes that encode innate immune response modulators, such as in the replication complex NSP1 or NSP14, or evolved in replication, the NSP14, NSP16, and then non-essential sequences here in the spike. Similar to what I showed you, the N-terminal domain can attenuate the PEDV if you delete that, and then some other deletions. And also deletions here in open reading frame 3. So the strategy is to introduce at least 2 distinct mutations into separate genes to attenuate the virus to increase genetic stability, but also for this virus to recombine with a wild type, it would take, if you do 2 or 3, it would take double or triple recombinants, which would be very rare to happen. But one can also put in mutations that will cripple the ability of this virus to recombine. So then the challenges for COVID-19 vaccines, they need to be rapidly deployed in terms of using these nucleic acid or viral vector vaccines. So these may be a first generation vaccine, at least to reduce mortality in the high risk groups. So in the animal studies, they generally in non-human primates, the chimp adeno Oxford vaccine did not prevent nasal shedding, nor did the inactivated vaccine. So they may also require booster doses to maintain immunity if it's short-lived, like the common cold. And then we may need second generation, more potent or efficacious vaccines, such as attenuated virus, live replicating vectors, better adjuvants, to actually prevent both severe disease and also to reduce or prevent shedding to block transmission. The other problem is many vaccines have reduced efficacy in the elderly or those with chronic disease. So the example is like for flu, which requires higher doses or better adjuvants or multiple doses. And then the concern is that the animal models may not reflect the vaccine responses in these high-risk groups. And at least in the phase 1 and 2 trials, people over the age of 55 were not enrolled. And the other situation that's going to present for the COVID-19 vaccines, they'll be used in 2 populations. Some of the population that gets the vaccine will be naive, and others will have recovered with variable levels of pre-existing immunity. So it's possible that either the efficacy or even the adverse effects, such as antibody-dependent enhancement of immunity, could vary. So finally, there's a number beyond this, but just some general unresolved questions related to my talk. What was the origin of COVID-19? How was it transmitted to humans? And under what circumstances does interspecies transmission occur? Is there an intermediate host that transmitted the virus to humans? Will humans spill over into animals, create a new host reservoir for COVID-19? It's still not completely clear about the role of asymptomatic and pre-symptomatic infections in virus spread and transmission. Why is there enhanced disease severity in the elderly or those with comorbidity, but milder disease in most children? How does the virus pathogenesis and host response with immunopathology influence disease progression, severity, and outcome? And then finally, do aerosols or non-respiratory routes, saliva, feces, foodborne or incidental hosts, play a role in SARS-CoV-2 transmission? So last, I'd just like to acknowledge our funding, our lab support. Dr. Vaslova was a former postdoc and research scientist in my lab. She's now an assistant professor, but we work closely together on a number of these coronavirus projects. Dr. Chahong's lab, who is pioneering the work on PDV infectious clone, which she has produced. She's also produced porcine delta coronavirus infectious clone, and now she's working on a COVID-19 infectious clone. Dr. Hu is now a postdoc in Dr. Barrick's lab, and he's actually produced a COVID-19 infectious clone. And then Dr. Scott Kinney's lab, who's working on the porcine delta coronavirus. So I'd like to thank you for your attention, and if there's time, I'll take questions.
Thank you, Dr. Saif. That was a very informative and, I must say, somewhat sobering presentation on our prospects for vaccines. One of the questions that has come up, and it relates to work that I know you've published on, I didn't mention at the beginning, you have several hundred publications on these RNA viruses, corona and rotavirus, calicivirus, including co-infections. And so now we're coming into flu season. You've done work with porcine respiratory coronavirus. Can you comment on how that might shake out?
So that's in part of the talk I didn't present about co-infection using PRCV as a model and co-infections between PRCV and influenza, which was, most of these were done in Dr. Van Ries's lab in Belgium. And we did co-infections with PRCV and PRRS. But the take-home message from these co-infections was the pathology, the shedding, sometimes loss of weight, were all more severe in the co-infected animals versus the single infection. But the timing of the two infections in terms of the disease presentation and pathology was very important. So interesting, in the influenza study, Dr. Van Ries found that the co-infections could actually reduce the influenza replication, probably through the inducing interferon. But in spite of that, the pathology was still more pronounced in the dual infected pigs than the single infected pigs.
Yeah. Wow.
Okay.
Well,
And
Dr. Saif, oh, go ahead.
I'm sorry, I don't know if Jeremy will talk about this, but the same thing with the bovine respiratory disease complex. So we started studying a lot about the bovine respiratory coronavirus and the shipping fever cases. And in most cases, if there was a co-bacterial infection, the disease, of course, was much more severe than just the virus alone.
Sure. Well, we are incredibly fortunate that you joined us today. We are even more fortunate that you work with us in the swine industry and beyond. And so thank you very much for your presentation today. And we hope Jeremy will get a chance to cover some of the co-infection issues with the bovine coronavirus presentation. So thank you very much, Dr. Saif.
Thank you.
Okay. So we will move forward to another presentation. Dr. Jeremy Scheffers is another one of the diagnosticians here at the Veterinary Diagnostic Lab at the University of Minnesota. He did his DVM training here. He was out he's originally from a dairy practice in central Minnesota, practiced in a dairy practice in central Minnesota before coming back in 2003 to pursue residency in pathology and then graduate school training where he did his PhD here on BVD virus. Dr. Scheffers I characterize him as a bulldog. He grabs onto a problem and won't let go of it. And I have one quote from something he wrote related to coronavirus. It's bovine respiratory disease complex, not bovine respiratory disease easy. Is that an accurate quote, Dr. Scheffers?
That is. It's complex for a reason.
Yep. So thanks, Jeremy, for helping us today. And he'll be presenting on bovine coronavirus mechanisms and vaccine considerations. So thanks, Jeremy, for helping us out today.
Okay. Hopefully I can share my slides here. There we go. And I think you guys have it controlled from there. Is that right? No? Let's try this. There we go. So hopefully you can see that. Yeah, thank you, Dr. Torson, for the introduction. I guess I won't elaborate too much. We're a little bit behind schedule. So I've been exploring bovine coronavirus for about a decade now, and the reason for that is when it comes to bovine respiratory disease, we were missing something. Robust vaccination programs in high health herds were still breaking with disease, and we were clearly missing something. Around 2010, developed a PCR test for bovine coronavirus, started testing lung, found lots of coronavirus in lung, didn't really know what that meant. So started digging into it. So just to back up a little bit, where does bovine coronavirus fit in the grand scheme of things? Bovine coronavirus in this table from Perland and Netland, it's an NIH document. Bovine coronavirus is, in this document, is a group 2A. It falls in the same group as some of these human cold viruses, and then HEV, porcine hemagglutinin encephalomyelitis virus. They all fall into this group 2A. And there's a few things there that kind of spike my attention. The one is, for the porcine one, we know that HEV has a nerve component. And And that to me, that's very interesting. When I read the virology textbooks, they talk about coronavirus having nerve involvement. So there's clearly some systemic issues going on there and a lot of those things have been, especially on the nerve side, have been poorly described. But I like this table because it clusters the human cold viruses with bovine coronaviruses. And I use that quite frequently. And I just tell people, what does coronavirus do to cattle? And it gives them a head cold. Just for your information, SARS is, Jerry's popping his head in here. Are we good? Slideshow button. Okay, can you show me where that is?
Nope.
What's that?
Must have done it already.
Okay, I think I did.
Okay.
Thank you. Anyway, so SARS-CoV-19 is right below that in a group 1B with obviously the bat and human viruses that Linda went over. So that's kind of what we're dealing with. PED and TG are above that. Feline infectious peritonitis is up there. So one of the things about coronavirus is that I have to remind myself and others, and especially veterinarians, is that coronaviruses do not stimulate interferon. For respiratory disease in cattle, we really like to give intranasal vaccines and stimulate interferon. We know the benefits of interferon as an antiviral compound, but I think it's important to remember that coronaviruses don't stimulate interferon. Coronaviruses are directly infectious. They move very rapidly. They don't stimulate interferon. And in addition to not stimulating interferon, they also antagonize interferon. In the group 2B, in the group 2A coronaviruses, there's mouse hepatitis virus. And they've shown with mouse hepatitis virus that not only is coronavirus is not stimulating interferon, they actually antagonize interferon. So that immunity feature might be one of the reasons why it appears that we can vaccinate in the face of maternal antibodies. It's rapidly replicating, and it completely goes around or goes through any interferon response. So for many, many years, growing up on a farm, watching calves scour and die from coronavirus, calves would be flat out. They'd have a bloody mucoid diarrhea. The picture above shows what feces looks like from a coronavirus calf. It sloughs a lot of its colon. There's a lot of mucus with that. There's some blood flecks. What you can't see is the very watery diarrhea. What you also can't see is a lot of bicarbonate loss. And a lot of those calves die of acidosis. So noting the mucus that's being sloughed out of the colon, I also know that there are goblet cells.
Sorry, one more that you're not on the right screen. Can you show your other screen?
Oh, I got to be on this one?
Yeah.
Is that working? That's working? All right. Fabio says that's working.
Okay.
Fabio gave me the thumbs up. All right. So we'll just back up a little bit. This is the chart. Again, human coronaviruses are clustered with some of these cold human bovine coronavirus and HEV. Coronaviruses do not stimulate interferon. Coronaviruses tend to antagonize interferon in calves and in adult cows. So it's calf diarrhea, adult cow winter dysentery, very similar to TGE and PED in pigs. There's a colonic involvement. They slough a lot of their colon, a lot of mucus gets sloughed. The bicarbonate loss is lethal. And knowing that and seeing that repeatedly, it draw me to think about, well, what happens in the upper airway where we also know there are lots of goblet cells. So we started, drifting away from the lung and looking further upstream. And upstream, you're going to have nasal turbinates, you're going to have tonsil, proximal trachea, you're going to have the middle ear, and all those structures contain goblet cells. So if coronavirus likes goblet cells in the colon, why wouldn't coronavirus like goblet cells in the upper airway? And it turns out that coronavirus does like goblet cells in the upper airway, and we can see replication of coronavirus in and around and near the goblet cells of the upper airway. So when we look at the tissues under the microscope with just regular H&E staining, we would say that those cells are attenuated. They're smoothened. There's not a nice columnar appearance to them. There's a loss of the cilia. And when we put IHC stains on them, they light up. And a lot of those, the lining of a lot of the upper airway is positive for bovine coronavirus. So it's not nearly as erosive or ulcerative or necrotic as a herpes virus infection might be, but there's clearly some replication happening. One of Dr. Saif's many chapters in many books is in the veterinary clinics of North America. A lot of the discussion in the bovine industry has been about, is the virus in the intestine different than the virus that we're pulling out of the lung and the upper airway? And the short answer is no, it's not a different virus. Short answer is no, it's not a different virus. I got 3 screens I'm looking at, and I don't seem to be advancing the right one. So this is the slide, sorry about that. This is the slide about the upper airway. Dr. Saif's chapter in veterinary clinics in North America, lots of words here. Basically, I'm boiling it down and saying there's really no difference in the virus that causes diarrhea and the virus that is found replicating in the upper airway. And I want to point out that the virus is not replicating in the lung. I don't see any type 2 pneumocyte lesions. I don't see any lesions in the distal airways, in the bronchioles. Everything is happening in front of the lung in the goblet cells. A study was put out in 2013 trying to decipher, are the respiratory coronaviruses in cattle different from the enteric coronaviruses that cause Widner dysentery and calf diarrhea? And the short answer is no. Across all clusters of the respiratory, winter dysentery, and vaccines, they're all 98.5% similar. So across everything, including the vaccines, they're 98.5% similar. So I approach the problem as we're just dealing with, thankfully, we're just dealing with one virus with 3 different clinical presentations. When we look at bovine lung that's submitted to the lab, bovine coronavirus is the most commonly detected virus in diseased bovine lung, and that's 25%. So cattle are not pigs, thankfully, and we don't have to deal with PRRS and Serco and influenza viruses. But when we just examine diseased bovine lung that comes to the lab, bovine coronavirus is the first virus that shows up. And we're not doing anything about it. We're just letting it run wild. So that, to me, is an issue. That's an opportunity to do something about it. So what does bovine coronavirus do in the respiratory tract? And there's nothing in the pathology textbooks beside me. There's no papers written. There's no papers written. And we're just kind of lost as to what it does. The first thing that I did is I started taking heads apart and again, looking at the goblet cells in the upper airway. So here's an example of a feedlot animal that died of bovine respiratory disease complex, and Mannheimia hemolytica bacteria was isolated. This one?
Yeah.
This one? Okay.
Yeah, there you go. Yeah, if you change that one, we can see. There you go.
Okay. Sorry again, there's 3 screens and I don't appear to be advancing the right one. So anyway, back to the pathology of what does bovine coronavirus do in the upper airway. It appears to replicate in the goblet cells. So again, we're going to have a rhinitis, a tonsillitis, proximal tracheitis, and wherever there's a goblet cell in the upper airway, we're going to have replication. So here's a bovine tonsil, and you can see that there's attenuation of the respiratory epithelium. There's a loss of cilia. There's a loss of goblet cell. There's a big blood pool forming. That's a lesion. Well, what's causing that lesion? Immunohistochemistry allows us to document where coronavirus is replicating. So same tonsil with attenuated epithelial cells, and lots of bovine coronavirus staining. That's bad in the nose and the tonsil and the upper airway because that basically destroys the mucus mat. We need the mucus mat to trap debris and have the cilia sweep it out of the upper airway. But in bovine coronavirus infected cattle, the mucus mat is lost, and basically we turn the upper airway into a slip and slide. So all the commensal bacteria, the Pastorellas, the Histophiluses, the Mannheimias that are colonizing the tonsil, in my impression, have a free trip down to the lung and cause bacterial bronchopneumonia. There's some nice studies done. Bovine coronavirus in the feedlot. Well, what does the feedlot give coronavirus? It gives the coronavirus exactly what it wants. It wants to commingle in confinement and have constant contact with other cattle. And so that's the feedlot setting. And in the feedlot, we've seen that bovine coronavirus is highly infectious. Both nasal and fecal shedding peaks 4 days post-arrival. On day 1, 11% of the cattle had antibodies, but by 3 weeks, 91% had seroconverted. It's a fast-moving virus. And of those cattle that seroconverted, it was inversely related to average daily gain. So the naive cattle that go in and seroconvert did not grow as well. So again, this shouldn't surprise anybody who's worked with coronavirus that it's highly contagious and it affects gain when naive animals seroconvert. Another nice study, it's 20 years old, but it just, to me, it rings true. Every one of the dots on that graph represents an animal, and the red dots are mortalities. Those are cattle that died. And then the blue diamonds are cattle that were asymptomatic cohorts. And the asymptomatic cohorts walked into the feed yard. So they walked into the confined commingling event with a coronavirus titer, and they were fine. Whereas cattle that walked into the feed yard with no titer had a much higher morbidity and mortality. So for me, it's pretty obvious. You want to walk into that confinement event with a titer. It shouldn't surprise anybody, but we have to ask ourselves, can we do something about that? We can just hope for natural exposure, which doesn't happen, or we can intervene with a vaccine. So to me, this is the justification for vaccinating cattle prior to the commingling, confinement, and constant contact event. One of the other questions that I wanted to try and decipher was, well, how long is immunity? I grew up on a dairy farm, and every winter we had winter dysentery, every winter, which tells me the immunity can't be more than a year. But then again, how long is it? So in some dairy herds, they have winter dysentery coronavirus infections in early November. And then they have it again in early April. They have it twice during the Minnesota winter. So that tells me that it's clearly less than a year, and it could be as short as 6 months. So what I did in a dairy herd is I just went through and I did serological profiling. What do the newborn calves have for a titer? Well, they have nice high titers from colostral antibodies from their vaccinated dams. And these 3 calves had titers of 640, 320, and 320. Then I went to the 10 of the 5-month-old calves, and their titers were decaying away. 3 of them were seronegative, and 2 of them had titers of 80. So these calves, not only did they get colostral antibodies from the vaccinated dams, they got an oral coronavirus vaccine, which oral vaccines and intranasal vaccines cannot be expected to generate a titer. So it's summer, and coronavirus doesn't thrive in the summertime, and these calves, their antibody titers are decaying away. And the older cattle, so here are yearling heifers, July of 2016, they were seronegative for coronavirus, and they were expected to be. Move over a few pens and we look at the springing heifers. Again, middle of the summer, their colostral antibodies have long since decayed away. There didn't seem to be an exposure event where they would seroconvert, and all these springing pregnant heifers are seronegative for coronavirus. But they're sitting ducks. They're seronegative, and as winter rolls around, they're just like pregnant gilts that get introduced to the sow farm. They're going to go into a confinement event and get commingled with older cows. and have constant contact with other cows, and they're gonna get winter dysentery unless we intervene and vaccinate them. And I can't tell you for sure whether or not that happened. So if you look at the serological profile, the lactating cows, or the sow farm, if you will, they should have a variety of antibody titers. The newborns should have a nice high titer, but in the case of bovine coronavirus, things start to decay away and turn totally seronegative by 6 months. So intervening with a vaccine. In the bovine industry, we have 5 vaccines to choose from. We have 3 killed vaccines and 2 modified live vaccines. And this is a farm that I had been working with since 2010, where every time he pulled his calves out of the hutches, he would confine them in a barn and commingle them with other calves, and they'd have constant contact with each other for a few days. And about 4 to 5 days after grouping, they would back off on feed. So they were eating about 3% of their body weight, and they would back down to 2.5% of their body weight. He would start to hear non-productive coughing. The calves were somewhat lethargic. Some of them shook their heads. Some of them had clear nasal discharge, and we would nasal swab them, and they would be loaded with low CT values of bovine coronavirus. So this is a picture of the barn from the inside out. This is actually a little better picture. This is a nursery barn for cattle. They're weaned off of milk, and they're confined, commingled, constant contact, and there's 200 calves in that barn. So before introducing them to the barn, we started a variety of different vaccination strategies. One of them was let's give 4 pens or 80 calves a sub-Q dose of killed bovine coronavirus vaccine. Then on the other end of the barn, we're going to do another group of 80, and we're going to give them intranasal modified live. And then we're going to have 2 negative control groups in the middle. And bovine coronavirus, the upper respiratory component of it, is a mild infection. It's just a head cold. And there were not a lot of morbidity events. There were no mortality events. So we really only got stuck. Our only outcome that we could measure was how well did they grow. And the calves that 5 minutes. All right. The calves that got the sub-Q killed vaccine, they did the best. The calves that got intranasal modified live, they did second best. And the negative controls were about 25 pounds lighter. They just didn't feel well. They didn't eat as much feed as the vaccinated calves. But again, the sub-Q killed calves did the best. So I remember driving away from that study thinking, you know what, I wonder what modified live sub-Q will do. And for the next group of 200 calves, we all gave them sub-Q modified live vaccine when they were 1 week old, and then again when they were 5 weeks old, and then they got grouped when they were 8 weeks old. So they all had 2 doses of sub-Q modified live. Those calves did great. They gained like gangbusters. They never backed off feed. We did a bunch of nasal swab trying to look for coronavirus, and we did not find it. So if we look back on that 5-year experiment, if you will, with coronavirus vaccines, we saw an improvement with a sub-Q killed, but we could still find coronavirus on nasal swabs. What shut it off, and when I say shut it off, what stopped, what produced negative nasal swabs was when we switched to a sub-Q modified live vaccine. And in this case, it was a calf guard vaccine. So we're able to go into that farm, vaccinate prior to the commingling and constant contact event, and we're able to build herd immunity and shut it off. Another farm that I dealt with in central Minnesota was a farm that had beef cow calf pairs. So this is a sow and farrowing crate situation where cows would drop their calves in confinement, and they would stay in confinement for three months. And these calves, even though they had antibodies to coronavirus from their dam being vaccinated, they still developed disease at about 3 or 4 weeks of age. So even though they had a titer, there was overwhelming exposure, and they still got sick, and there was lots of coronavirus found on nasal swabs. This case scared me a little bit because I just don't think we could build immunity in time, and actually we were able to. So you're looking at the calves, but just appreciate that the cows are all on the other side of the calf area. And we were able to vaccinate these calves on day 2 and then again on day 12. And we were able to build immunity and get enough herd immunity going in the calf population that the quote unquote coronavirus fire never started. So we were able to vaccinate in the face of maternal immunity and stop a disease in this confinement event. In addition to confinement and constant contact and commingling, it was cold out, which coronavirus likes, and it was cloudy, which there was not much UV light desiccation. So I've had some success vaccinating for coronavirus in cattle populations, but there's some rules. I'm going to wrap up because we're just a little bit over time. So my summary for bovine coronavirus is that they are directly infectious and highly contagious. They do not stimulate and can antagonize interferon. For cattle, it seems to affect the goblet cells of the upper airway. We need to protect the upper airway to protect the lungs. So I believe protecting the upper airway is a good idea. I've had better success with modified live versus killed vaccines. 2 shots work better than 1. Herd immunity is very important. We appear to be able to vaccinate in the face of maternal antibodies. The vaccines that we have are safe. There are almost no adverse vaccine reactions. Herd immunity is critical. We need at least 80% of the herd to get some herd immunity. And from what I can see in the cattle population is serum antibody titers don't last longer than 6 months. And serum antibody titers can be overwhelmed in the face of overwhelming exposure. So I think it's hopefully a little bit of good news on the vaccine front that in one population or one species of animal, we appear to have made some headway and just try to understand the idiosyncrasies of how this virus behaves in cattle populations, and maybe we can extrapolate that to other species. So with that, I'll turn it over to Jerry if I look at the right screen and push the right button.
Jeremy, I don't think you have the button, Jeremy, but thank you very much. In Linda Saif's recent article from last month, she mentioned something I hadn't realized. A couple of times in the 1700s, 1800s, ruminants passed a bovine coronavirus to humans that apparently are among the coronaviruses that are the cause of the common cold. So cattle and other ruminants aren't off the hook here either in terms of potential zoonotic disease. I think we won't take time to ask any more questions, but thanks very much. Sorry for the technical glitches there, Jeremy, and thanks for soldiering through. And so we'll segue over to our wrap up for this session. We've asked Dr. Rossow, Dr. Stephanie Rossow, another diagnostician here at the Veterinary Diagnostic Lab, to help us put it in perspective from the standpoint of the pig. Dr. Rossow, I don't think needs a long introduction, so I won't make everyone suffer through that, except to say that she's really been instrumental for decades here from the viral molecular and bacterial molecular standpoint. Those of you who recall the early days of the PD outbreak know that she and her group put together a PED vaccine and had it online in less than a week. And so we saw an example of how it can go when there's an epidemic that comes in. So Dr. Rossow, if you could take the comments from Dr. Saif and Dr. Scheffers and help us put it in perspective from the pig standpoint, that would be helpful. Thanks.
Sure. Thanks, Jerry. I'm assuming you can hear me, and can you see my slides?
We can hear you, and they'll switch to your slides.
Okay. Okay, can you see my slides okay? I'll take that as a yes. So I was asked to kind of wrap up about coronaviruses in pigs, and so with a little license, expanded that topic a little bit to proceed on. Can you see my slides? Because I'm clicking and it's not working on the screen. Someone let me know. I'm not going to say anything until you let me know. Okay. Or someone can just type me a chat and say it's okay. Okay. So I'm going to skip through. Skip through a bunch of this since my kids quit working here, and just briefly go. And the slides aren't advanced. And so basically, this is more talk on the coronaviruses on people are trying to understand what's going on. And it's become clear, obviously, the opinions and observations of the rest of our mind, not representative necessarily of anyone else's. But we don't look for disease thoroughly. We don't understand interactions of disease very little. And so people like to have that financial appreciate the opportunity we have listening on and so looking for new things and looking for old things. So the question comes up, PRCV, it's not around anymore. We don't see it. We don't see TG anymore. And why is that? Is it because of PRCV? Is it because of better TG biosecurity and it just went away? And the honest answer on PRCV is we don't look for it. And so it's hard to know if it's here on a routine basis. We pretend we don't think it's here based on what we see and what we don't see. But again, basically, we don't look for it. And the other coronavirus that I know is still here, if you look at it in literature, it's called uneconomically important, not important. It's called, it doesn't show up very much, and it's largely opinions provided by people that don't look for it on a routine basis, because if you look for it, you find it, and it's can take a little time to find or the right tissues to find, but all you have to do is look for it. And kind of like coronavirus in cattle, you can find it in the ethmotubernates, turbinates, you can find it in the lung, you can find it in the tonsil, and you can find it in the stomach. And I base those on looking for it in those tissues individually. By PCR tests, we don't have a way to look for it, stain for it in tissues. And so it's there. All you have to do is look for it. And like a lot of things, we just don't look for them. And we don't look for them mostly because it's expensive. And it's expensive in reagents, it's expensive in time for people, it's expensive in trained technicians, and the expense comes back from multiple levels, from clients, from university, and from state governments, and from federal governments. And of the people listed, generally the people most amenable to spending money looking for things are clients. And so that's always been one of the pluses for me working with pigs, is that clients, swine clients, are almost always interested in looking and trying to do better on that. But again, we don't look for things because it's expensive to look for them. And one of the justifications that you can come across is sometimes it feels like you have to prove something is important before you can prove it is important. And so there needs to be opportunities to just look for stuff on a routine basis. And so it'd be nice to be able to look for things, lots of things, everything on a routine basis. And currently we can't do that. We have next generation sequencing, which has been a great benefit on several accounts, different opportunities to find opportunities. But from using it and spending money on it, I know it lacks sensitivity. You can miss stuff with it. It has biases for amplification, so you can miss stuff that's there. It's expensive and it's slow, and I use this in sense of using it as a routine diagnostic test to provide timely answers and immediacy for people, which is what people want. And so also part of this consideration, when you're looking for stuff, we're thinking about trying to figure out what's important. You have to think about all these things historically too. You go back in literature and say, well, they gave pigs this virus and it caused this disease, caused these lesions, and we know that because we checked for other things by virus isolation, and nothing else grew. Well, we know for now, we know now that's not a sensitive way to see what other pathogens may be co-circulating in your experimental animals or in your natural infections. And used for a couple examples for that, growing up as a pathologist, you taught a lot of things, and one of them was that Haemophilus pyrrhusus or Glasserella pyrrhusus was the sole cause of polycercitis and arthritis in pigs. And Mycoplasma hyalurinus was a non-event, wasn't important, and don't waste your time looking for it. And so we learned, once we had the opportunity to apply PCR to routine polycercitis diagnoses, in that it is important, it is there commonly, and it is a significant pathogen of pigs. Another example, I can give an older example that one of my advisors always threw at me was soybean meal causing diarrhea in post-weaning pigs. The caveat on that is, if you go back and read the papers on that, you'll quickly see that they didn't test for any infectious agents in post-weaning pigs that were commingled. And so anyone today should know that when you commingle pigs post-weaning, you're going to have rotavirus at least in those pigs. And so when you go back and think about what your biases are, and saying, okay, we know this disease does that because long ago they reproduced it, that doesn't necessarily mean that was well defined or thoroughly defined, or defined as well as they could at that point in time. Another consideration that for me when looking for things is that sequencing has been a great boon, great plus for understanding epidemiology of viruses, who's getting what, where they're coming from, how do you keep them out. But we need to advance beyond that. Again, it's a matter of resources, getting committed, committing people, training people to routine diagnostics, and try to use that sequencing information to establish a relationship to immunity day in and day out on things, that information that we generate, because that's how people learn things That's how clients learn things. That's how vets learn things, is to see them day in and day out, so they can make those clinical associations with diseases that we're finding for them. Looking for things continued. Tissue lesions. We like to find bugs in tissues so we can define what cell types they're replicating in. And I'll try to close out here shortly. And it's some consideration on something as simple as looking at a section of tissue under a microscope that's half the size of a postage stamp and thinner than a postage stamp, as opposed to a PCR test on tissue that's got several chunks of tissue in it the size of certain sugar cubes. And so you're comparing somewhat apples and oranges on trying to understand necessarily what you see under the microscope and what you find by PCR test. And so I'm a histopathologist by training, and it's a great and powerful tool, but you need to keep perspective on what you're looking at and trying to understand between different tests, and setting the bar for what you see just on your histopath or just by one technique is really going to limit your opportunities to have a positive effect on pig health. And so to me, what the future for now and anyway is going to be looking, what I would really like to do is to have multiplex PCRs for multiple pathogens. I'm not going to go through these other than to demonstrate that people are finding ways to look for 16 pathogens, 17 pathogens, anymore. I love this title, Seek and Find. If you look, wow, you find stuff. And you have to look with the right methods that are sensitive to pick up endemic disease, that aren't going to be present in extraordinarily high numbers. Just quickly, as a quick example of I'm really not exaggerating on looking for multiple diseases in pigs or finding multiple agents in pigs. 3-day-old pig remains a 3 15-day-old pigs. I'll just go through here quickly and concentrate a little more on the older pigs. Little pig did have a nice group C rotavirus. Pigs with tremors, finding atypical porcine pestivirus, finding prior enteritis in those pigs. We're finding PCV2, finding nutritional disease issues on the growth plate. You look at the older other pigs with tremors, finding atypical porcine pestivirus, HEV in the stomach with lymphocytic neuritis, got acute arthritis and meningitis with strep suis. Similar with the other pig, arthritis, neuritis, PCV2 infection, although no evidence of disease. And so just a quick example that these are everyday events. These multiple agents are everyday events. If you want to understand them, you need to look for them. And so what I'd like to do is shift to more routine multiplex PCRs. You have better sensitivity, you have faster turnaround time, they're less expensive, and they're compatible with the current equipment. And then also add routine structural profiles to sequencing events to get a better idea on antigenicity for routine cases. And with that hurried presentation, I'll quit. Thank you.
Thanks, Dr. Rossow. We didn't give you much time at the beginning, and then we cheated on it anyway. And so thanks for drawing us together in terms of some thoughts on coronaviruses and other viruses and some diagnostic approaches. This concludes our coronavirus session. I want to thank Dr. Saif, Dr. Scheffers, and Dr. Rossow for their presentations. We'll try to circle back with responding to some of the questions that came up in the chat room. And for now, please enjoy your break. Visit the sponsor tabs, and we'll see you throughout the rest of the conference. Thank you very much.